Hello, everyone. The Center for Integrated Environmental Health Sciences and Department of Pharmacology and Toxicology welcomes you to our Environmental Health Sciences series seminar. Our speaker for today is Dr. Ted Smith. He received his bachelor's in biology and also psychology from Allegheny College and master and Ph.D. in environmental psychology from Miami University and postdoctoral studies at MIT. Dr. Smith is an associate professor in the Division of Environmental Medicine and director of the Center for Healthy Air, Water, and Soil at the Christina Lee Brown Environment Institute at U of L. His research focuses on environmental technologies that provide place-based clinical insight for preventing or reducing the burden of disease. Dr. Smith was previously the Chief of Civic Innovation for the city of Louisville and created the largest real-time asthma monitoring project, Air Louisville. He also leads the wastewater monitoring on behalf of Louisville Metro Health Department. The project has been sampling SARS-CoV-2 from community sites across Louisville since June 2020 and sequencing for a broad panel of pathogens since February 2022. Dr. Smith also leads the Commonwealth of Kentucky's wastewater monitoring of correctional facilities for the State Health Department. He has also been a consultant to a CDC contract for Senior Living Center wastewater monitoring. He has published 26 papers in this field since 2020. His talk for today is Multimethod Approaches to Maximize Insights from Wastewater-Based Epidemiology. Let's welcome Dr. Ted Smith.
All right, good morning. First, my apologies for anybody who might have seen a version of this talk earlier this week, but it was a great pleasure to present some of this in brief to our external advisory committee that was here visiting for CIEHS. So again, my apologies. You'll ask me the softball questions because you know what's in it, but for everybody else, it'll be fresh and exciting. So maybe a little framing that I didn't have time to do earlier this week is in order here. So the talk is really focused on mixed methods. And when people use the term mixed methods for research, often they mean coupling quantitative research with qualitative research. So I might have some, robust quantitative information, but I'm missing some of the color of what's going on. And so I would complement that with narrative. But the strict definition of mixed methods is combining more than one method together. And so quantitative and quantitative is a perfectly appropriate design. And so, while we're going to talk a little bit about COVID and we're going to talk a little bit about metals, and we're going to talk the focus really of the talk, and I would like you all to think about this as a platform. Think of it as a facility, a research facility model. And, can I turn to this platform to answer questions that I have, whether they're basic research questions or applied public health questions. So I'm going to ask you to try to keep your brain in that place, because that's very much the way we've been looking at it, going back to 2018 here at U of L. And for me in the city, we first engaged on this in 2014. When we were approached by Arizona State University to look at opiate surveillance in the city. And so it's a young field in the United States, and it has really only proven its value in infectious disease on scale, and thanks to the pandemic on big scale. But it is a young field, and I encourage you all to think very broadly and ask very difficult questions about it. Okay, so if you like environmental monitoring, you probably like the idea of having an array of monitors that can monitor something in the environment. Or maybe you like to have a highly described clinical cohort, that's, very representative of a place. I've come at this from environmental monitoring. And so the asthma research that I've done in the past was focused on heat maps of places and people having asthma attacks and looking at air pollution data and trying to figure out what those relationships were. So when we think about the wastewater system, you have a a highly developed infrastructure that is population adjusted across all the cities in this country. In fact, any town with more than 10,000 people in it will have some kind of sewerage system for eliminating waste. And so this is Jefferson County as seen under the ground. So under our feet today is some portion of that map of pipes. And it is well described in the logic system that many have used to do various kinds of research on the epidemiological front. We look to maps of the city where people live, their demographics, all those kinds of things, layers and layers in ArcGIS. Well, one of those layers is the sewage system. Okay, so we can look at places and we can go into a GIS platform and try to figure out if I can isolate from the sewage a certain place inside of our county. Okay, now this is just an important thing to get out on the table early on. That one of the reasons that NIH, I believe, has struggled with this as part of a research program historically is because it is not a clinical sample. Okay, it is garbage, and so many of the reviewers, use language like that, which is helpful to remind us that it is a system that really handles waste, right? Now, in that waste happens to be biospecimens, and not only human biospecimens, but all kinds of other organisms, right? So there's a whole biome in the sewer, and there are many different kinds of contributors to that biome. And there are many sort of secondary reactions, right? So just like your gut, you're also feeding a whole system inside the sewer, right? That's making its own products and is using its own inputs. So, the full description, we haven't had Mike Snyder lately go in and show us the Galapagos inside the sewer, in this big style of sequence everything you can find. But it would be fascinating to see what the whole thing looks like because it's very complicated. Now, that's one problem, that you're looking for a signal and a lot of noise. I mean, another problem is it's not really a research platform, right? So when we go to the IRB, and we've done it a couple dozen times now, and said, hey, we're going to now go look at this, or we're going to go look for that. Every time I get a letter back that says this doesn't meet the common rule definition of human research, and for good reason. It's every time we go do this work, we're not looking for any one person ever. And so we're always aggregating a population of people. It is by definition de-identified if we don't start hunting for discarded human DNA, which we told the IRB we will never look for discarded human DNA in our work. But of course, conspiracy theorists, have their ideas about should we have our sewage surveilled. I've gotten threat notes in my past. So just know that that doesn't typically happen in a clinical trial. Hate mail doesn't come from the public for those activities, but this is kind of a gray area, right? Because it was really designed to handle garbage. It wasn't designed to do research. Knowing that, is there any promise? Well, let me get to promise in a second. We have to do the tutorial. How does it work? Because I know we're all curious. By the way, we're building an exhibit at the Kentucky Science Center on this area. And so I know we all have like an inner 6-year-old that really wants to know how the sewer works. So just go ahead, get ready. So it's a gravity system, generally speaking, in most cities in America. And so the whole idea is the sewage, falls from your toilet and falls into the ground and keeps falling on its way to a treatment plant. And when it runs out of falling, a pumping station appears, which pumps it back up and gives it some pressure to keep moving. And so there are many opportunities in a system that works like that to get samples, right? One of them is the obvious one, the manhole, right? We all walk around them all day long. And so, we can open one up and we can, grab a little out, and we've done that for lots of projects, and we go and analyze what we found. We can hook up a $14,000 24-hour composite sampler, and we can get a 24-hour representative picture of what's going through those pipes. Or we can go to one of our 5 wastewater treatment plants in Jefferson County, and we can essentially, I think of it as like a Guinness tap. You just pull the tap, fill the sample jar, and go back to the lab. It's the easiest way to get a sample imaginable, but it is also probably the least spatially representative because you're aggregating hundreds of thousands of people in most cases. And so, we're getting a a Louisville answer. We're not getting a Chickasaw answer or a Linden answer. And so we have to think about our field methods. We can also think about it just in terms of aggregation. And so, there's a big difference between the kinds of things that you can learn about whole communities and what you can learn from a facility like a nursing home or a prison. And so our work has spanned these different levels of aggregation, and we've tried to understand the relationship when they're nested in each other, as they always are. So, that campus could be inside one of the large neighborhoods, which is inside a larger catchment area. If we have a pathogen or a toxicant of interest, we might want to see what the strength of the signal is in each of those aggregations, right? So it's an important thing to think about as an opportunity. This is how we sort of boiled it down to sort of stay sane. In the software industry, it's called a V model, and the idea is anything that's on the left has a kind of a reaction or an action on the right. And so if we're thinking about the whole community, what can we really look for when we're looking at the entire community? We can only really look at trends of things over time. What can we do about it if we found something disturbing? We can only really do sort of broad kind of policy things for the whole community. As you get higher levels of resolution, we have more opportunities for interventions or policies, right? So we get to neighborhoods, and we have a lot of experience working in neighborhoods in Jefferson County. We can look at differences between neighborhoods, and if it was COVID, we'd say there's not enough testing going on over there. You need a vaccination program over there. Whatever it may be, like, we can direct traffic if we have that kind of resolution. Obviously, once you get down to a nursing home or a prison or a college dorm, you're in a very different ballpark, right? Where you can perhaps know everybody that's in the building, and you can perhaps look at, screen, test, offer an intervention to the defined population that's in that campus, in that facility. And so it's a completely different level of insight and a completely different level of interaction. It is also a completely different level of cost. So as we go down here, it gets more expensive. So when people say, yeah, it sounds like a no-brainer to do infectious disease surveillance at senior living centers, it is a big brainer because we have thousands of them in the country. And to implement this kind of sampling infrastructure would be very, very expensive. So much easier to go to the wastewater treatment plant, pull the tap, fill the bottle, and go run a sample. So, when people say, well, wastewater surveillance or wastewater monitoring is so cost effective, we have to be kind of clear about what we're talking about here, because it may or may not be. Or maybe the cost is justified, because the benefit of a C. diff infection in a nursing home is, catching it is a really valuable thing. So, okay, so when we get back to methods, right? So we've got this platform, you know how it works, you know how we're supposed to go get samples and how we think about it. Then we said, well, how do we actually do research with this? And so in COVID, it's obviously an easy place to start because there's a lot of familiarity, and we have a whole lot of data, and we've done a lot of work in this area. It has often started with, tell me what the wastewater says today. I still get calls today. We have 500 people who go to our dashboard every week on the sewage data today, right? The pandemic's over, right? The emergency's over, right? Like, we still have 500 people who want to know what the wastewater says about COVID today. So, and they look at it, and then I don't know what they do. They panic sometimes, maybe they get comfort sometimes, but I don't know what to do with that, right? All I can tell you is there's COVID in our community. I don't know what it means. I don't know if that's a lot of COVID. I don't know if that's a concerning amount of COVID. I don't know if we should call the hospital and let them know they're going to have a lot of patients soon, or buy another ventilator. These are the kinds of things that I think we would want to know, and when we look at data, but we don't know them. Now, most of the country spent the pandemic looking at their wastewater and then looking at test results, right, back when we were doing testing all the time, or they were looking at hospital statistics. So here you can see just before the CDC turned off the hospital reporting, which was a month ago, we were routinely running analyses like these, which essentially the blue is the wastewater levels in Jefferson County. And red is, at the top panel is admissions with COVID, and the bottom panel is percent ICU beds with COVID. And so you can see wastewater, I mean, it's pretty jaggedy, right? We've got to really understand how much variation there is in the signal. But, the wastewater goes down, the hospital data goes down, the wastewater goes up, the hospital data goes up. The slopes aren't the same in all cases. The timing, there's a phase relationship, right? We learn about it in the sewage before we see the cases in the hospital. So this sophistication about how much of a delay we should expect is an important matter, right? If you want to inform public health, you want to have a good facility with that. But those clinical data are not available today. And so it's getting a lot harder to answer the question, should we be concerned, right? Because I think we should be concerned if hospitalization was going up, if ICU beds were going up, and if we knew that the sewage was giving us lead time, that we would be having that kind of conversation. But again, if you don't have that other data set, it is impossible to speak intelligently about what the situation is, other than to say we have an endemic pathogen in our community. It doesn't seem to be all that seasonal at this point in time, and that's all I know. And we continue to monitor it, because if it takes a left turn and we get some crazy mutation, maybe we'll be glad we're just keeping track of it, right? But I mean, in terms of a research platform, COVID's kind of worn out if I don't have something else to look at. Earlier in the pandemic, we did an ambitious seroprevalence study to try to answer the question, can I figure out how much sewage equals how much prevalence of infection? And we couldn't do that with the administrative data, the hospital data, because, or the testing data that was available from the county, because there was under testing, right? We knew that the whole time. That was before home tests, and then it was a disaster at that point in time. We could never know how much COVID there was in the community from the administrative data that was available because of the under testing. Now, there's also under shedding in feces, right? This is actually how the virus is detectable to us. So the endothelium in the gut is shedding SARS virus, and that shedding doesn't happen uniformly between people. And so we're going to miss something in the sewage, but I think we're missing a lot more on the testing and on the other administrative data. So what we did is we did a randomized, stratified random sample of Jefferson County residents, 5 waves, each with, 5,000 people in it. We mailed 30,000 postcards. Each wave, we worked with Westat to design the frame. It's a rock solid approach to survey. So we did active infection, we did antibodies, and we were then able to estimate reliably what the level of infection from the seroprevalence exposure rate has been, how many people have been infected. And then we were able to take the imputed, so we knew how many people were infected over time, multiple waves. We could then impute what the incidence rates were likely to be, and we could compare those to the wastewater levels over those periods. It's a really complicated model. Folks at Ohio State did all the heavy lifting on the basic epidemiological modeling, and we came up with a mathematical equation. Only people in the world to come up with a mathematical equation that describes levels of wastewater in terms of prevalence of infection in the community. But it required $3 million of randomized testing in our community. Not scalable, not clear that our formula, our ratio is the same as the one in Houston, or Dallas, or wherever else you want to look. So, an expensive, prove the point, but probably not a road that's going to get the country to a definitive way of understanding wastewater as the only source of information, right? Okay, so that's COVID. There's a whole bunch of other work in infectious disease. I'll touch on it briefly. But thanks to Chris, we've had an invitation to come over to toxicology land and leave our friends in infectious diseases behind for a little while. And so the question now is, can this platform help us answer research questions in toxicology, environmental toxicology? And it's a hard problem. We'll talk about that here. So our first foray, and a big thanks to Lou Kai and a bunch of other folks who've helped us, learn about new and other kinds of lab methods to work with the wastewater. We've done a broad survey sample of Jefferson County, 26 sites across the county, kind of weighted in our industrial core. And we said, gosh, if we wanted to understand what the metals look like, forget about COVID, forget about all that stuff, let's just look at metals. What we found was there's a gradient of concentration. This is the way I like to think about it. The first order of business is to, determine whether this is all just uniformly what's in the sewers or whether there are concentrations. And if there are concentrations of a toxicant, we might want to then ask the next question, why might there be an elevated concentration in this area? And we might want to ask other questions about what might some sources be and whether the likelihood that this is also related to a human exposure. That's much harder than the world of infectious disease, where SARS-CoV-2 was primarily a human pathogen in our community, not as much wildlife to be worried about or other kinds of things that might have gotten infected. So it was a cleaner frame to work in. This is going to be much harder, right? Because we have to reconnect the toxicant to the person. So, but I guess the good news, if there's good news, is the story at the highest level is rhyming. And so if we look at the toxic release inventory on the right and the metals in the sewage on the left, what you see if you squint is kind of the same thing. This is particularly true in West Louisville, where a lot of our industrial activities historically have been and still are. We know that there's plenty of different kinds of complicated toxicants that are released every day. And we see some of those elements in the wastewater in those areas. So this is an important first step. It's missing, as I mentioned earlier, some other reference point. So I could tell you about the gradient. I can't tell you about the health effect. I can't tell you about the source because these are things that we're going to have to go figure out. And so I'll just give you a sort of sense of what it looks like. I apologize for the small type, but this panel of 26 metals, we really just, the first order is just to see whether there were different levels of concentration. If I compare the county average for each of these metals, and I look at any location, I can see that there's going to be a range. There's variability in the levels of these metals. And I just showed you it on a map. There's variability spatially in these things. And so then the question is, what do these things have to do with health? Where do they come from? Are they exposures? I mentioned earlier that a lot of the easiest work to do is to look at these aggregation issues, right? So if I go to the wastewater treatment plant, which is fed by lots of those areas that I showed you, will I see the same concentration of metals of concern? And the answer is I will not. So I will see in some parts of our community higher levels than in the place they feed into, right? It's one big closed system. So that's another way I can see that there are gradients of concentration of metals because I've got a closed system with a 1 point and then an aggregator. And if I see the same ratio at the aggregator that I do in the point, then I say there's not a story here, but that is not what we found. So we found places higher lead, higher arsenic than we found in the place that aggregated the sample. Okay, so for this work, so toxicology, environmental toxicology, a lot of the work of CIEHS, I hope, is to start to move a little further in, right? And so are we looking at the whole range of toxicants? The answer is no. The EPA has a big concern about a whole list of emerging toxicants, and the fluorinated carbons are high on the list, so PFAS, PFOA, all that. And we're waiting by the post office every day for the results to come back in Emory on some untargeted work for these PFAS chemicals in our wastewater from those areas. We have to have a better handle on the industrial discharges into the sewers, right? Remember, it's in an industrial core. There may be industrial activities that aren't necessarily directly related to human exposure, so we need to understand that. It takes essentially an open records request to get that information, so it's not fast, but we will do it. Chris made a wonderful suggestion on Monday that I've incorporated in Friday's or Thursday's presentation, and that is really taking a look. I didn't mention it before, but everything inside the Waterston Expressway, generally speaking, is a combined sewer system that's handling stormwater and human waste. And outside of the Waterston are dedicated sanitary sewer lines, and then the stormwater is dealt with entirely independently. And so maybe we can start answering some questions just by looking at samples inside and outside the Waterston for potential environmental components that we might just be able to see in that contrast. It's a wonderful suggestion, pretty straightforward to do. So we will do that. Looking for the human health data is a big deal, and so we're going to have to think through whether that's administrative data, the wonderful claims data that I know we've worked with here, and other sources, anything out of the EHR from ULH. I mean, there's things we can do. We have options. And then, kind of one of the things that the community would like to hear is that if we find something scary, we're committed to doing something about it. And this is a lesson, obviously, I took away from the pandemic, that there's not a lot of patience for just research for research's sake in the community. Not a lot of patience. There's a history, a reputation of extractive research. Drive nice cars into low-income neighborhoods, do your study, drive your nice car away, and everybody says, well, we're still sick, right? And so I think this is a really important turning point for the work we do in environmental health, where if we can ever think of something to do, something interventional, we should try to think of that thing. And so with the sewers, we have an opportunity to say, well, if we see some elevated level of arsenic or elevated level of whatever, maybe we can figure out what's going on there. And if it is a health risk, maybe we can do something about minimizing it, and so, or eliminating it. So, I mean, this is what we really also need to think about this platform. This platform can help us find things and potentially do something about it, and then see that we did something about it, right? One good example of that is some early work with Dr. Dept and Cindy Kovalakis, who's a colorectal surgeon. They found 3 hotspots for colorectal cancer in Jefferson County. They're pretty tight spaces, like call it 300 households, 100 households, a couple of city blocks. And so, one of those hotspots on the left in the box is the hotspot where the folks live who have colorectal cancer. And the red polygon is the catchment area that we can isolate. That is just that area, no other contributors outside. It's pretty clean. The rest of them aren't so clean, the other 2. That's why I showed this one, to mislead you that this is easy to do. So there we have it. So we can get the sewage for that area. We can go do the metals analysis, which we've done here. I've circled the metals that have been demonstrated recently in the literature to be associated with early onset colorectal cancer. In almost all cases, what we pulled out of the ground in those hotspots are the very same metals that have been identified to be risk factors for colorectal cancer. This is giving us confidence that if we had even more data, and the good thing about Dr. Dept is she's a good epidemiologist, and so she's working on a case control methodology where we're looking at the blood of the people with colorectal cancer and a well-matched case control. Unfortunately for me, we don't have the wastewater for the case control people in the same place that we have as just a comparison for this. So we have some homework to do when we mix those methods so that when she looks at blood of these 2 groups, we also might have an environmental proxy for those 2 groups. And if we don't have it exactly neatly, we're going to have to do a little finessing. But I'm very optimistic that this is the kind of work that is not only advancing our basic understanding of these toxicants in the environment, but even to be able to do something about it is very exciting. Okay, so I've talked a little bit about sort of next steps on the metals. We got some good feedback from our critical friends in the external advisory committee on Monday. So I think they've challenged us to think very carefully about where these metals are coming from, how we might want to make adjustments, whether we need to divide everything by iron or not, or I mean, there's lots of interesting feedback. Dr. Conklin made the good suggestion that maybe we do some isotope research, and in the case of lead, we might be able to understand where the sources are of the lead if we do work like that. So, I mean, there's a lot of other questions and other approaches that we're going to want to incorporate and think about, and that's exciting. Related work, through the Superfund program, we're focused on volatile organic compounds. This is actually how we got into the sewers at U of L. Before the pandemic, we were interested in seeing if we could isolate urinary metabolites of these VOCs. We've done that. We've published that work. We've demonstrated there's an approach to doing it. It has its problems. Our good friends at the EAC were happy to remind us of some of those problems. We have to adjust for smoking, and so we do that with cotinine. We adjust, sort of a normalization. But then I think Dr. Cave brought some really good points about, are these the only sources of these compounds? And the answer is probably not. And so while I might be able to show you that there's differences in these presence of these metabolites in areas where we might expect that there would be, because there's more VOCs that have been documented in air toxics or whatever, the case is not simply closed at that point in time. So we have some work to do with this, but the attractive thing here is monitoring for air toxics is expensive. The Superfund program has a complicated Toyota Prius, that has an auto GC in it that has to get recalibrated every 2 miles. The air pollution control district has a thing that they can't calibrate. They've had it for 5 years out of the firearms training center. Otherwise, people were putting it in canisters and shipping it off, and find out about it 6 months later. So if we were able to get into some kind of system that was regularly monitoring metabolites of air toxic exposure, I think it would change the game in environmental justice communities to give them more tools than kind of the ones that are kind of expensive and clunky and slow right now. As one real practical example, you remember that we had a chemical hoarder down in Applegate Lane in Highview. So that house was discovered, sort of last spring, and in the summer, the EPA, all sorts of ASTDR, whatever, they all came in and they said, this house is a real mess. Over 300 chemicals mixed in ways that people didn't understand. A hoarder, so there was no safe way. A robot couldn't even get in the house to sort of pick through it. And so we got called in when the city said, I think we're just going to light it on fire. And so I don't think they had watched the East Palestine thing, because if you light something like that on fire, it will create a big, nasty black plume in the sky that everybody will pick up their phone and take a picture of. And then every plaintiff's attorney will appear in this town within 2 hours and begin enrolling people, right? So the craziness, in my personal opinion, of lighting it on fire put us into an immediate kind of a defensive posture. And essentially, I called up MSD. We took all that urinary metabolite stuff. I called up Pavel and our mass spec core. And we said, we will begin collecting sewage now in this area. And so you're looking at, in the colorful polygons, the catchment areas that surround, essentially, I think it's a mile of the house that was going to be put on fire. And we are just now emptying the freezer of the samples because the house has been dismantled, was not burned. It was picked apart by a long crane after they surrounded it with steel containers. And it is now officially clear. And so we've stopped pulling sewage, but we were ready to write to the Superfund program and say, here's a case study, natural experiment. Somebody, created a whole bunch of air toxics at once, and we were able to show you what wastewater was able to do. So, so I'm a little sad that we didn't have the case report. I'm certainly happy for our community that we didn't light the house on fire. But this is a real kind of up close and personal application, right? And I think for emergency response, disaster management, I'd like to think how these tools can be useful in that situation. Another application, and this is actually from the middle of the pandemic, we realized that if we knew that there were differences in testing rates because of undocumented, like elevations in the wastewater that weren't comporting with elevations in positive test results, that maybe we could create interventions we were featured in the only National Academy's report in this field, which came out at the beginning of this year. This ad intervention was the only interventional public health project in the United States, which is insane. So, and it wasn't really designed to be a big project. It wasn't funded independently. But what we essentially said is, if I know that there's a couple of neighborhoods that have really high levels of SARS-CoV-2 in their sewage, but I don't see any positive test data from that community, you know what I'm going to do? I'm going to get a geo-targeted digital ad, and I'm going to take a picture of things that are in your neighborhood, that neighborhood, so that when it shows up on your phone, like all those other junky ads, like if you live in Kentucky, your car could be for you, or whatever those things are, you know, people actually have the ability to pay attention to things that are familiar, right? And so we did that. We went to neighborhoods that had high elevated levels of infection, and then we said the wastewater says you need to get tested, you should get vaccinated. And so, and then we had control neighborhoods. We didn't run sham ads. That's why it was getting a Nobel Prize today. But it was definitely a good way to take that GIS data from the sewage geography and turn it into an intervention that followed the exact same borders. So here you can see at the top panel is our sort of our target community, you know, that had high levels in their wastewater. This is testing data. This is COVID testing data on the blue line. And so top panel is a community that we saw high levels of the sewage. The middle panel is a comparison neighborhood, same demographics, same size, but you know, not problematic in the sewage sense. And then the bottom panel is the difference between the communities. You can see we ran the ad campaign. I mean, I don't know if you know digital advertising, but you send 100 million digital ads out into the world in a very short period of time. And so you can see in this case, while not statistically significant, you know, we get encouraging trends. This is what we saw, that the neighborhood that got the ad campaign did have an increase in testing in the period that concurrently and in the period that followed. Now, I have a slide that shows you it also doesn't work, right? So I have a couple of neighborhoods, an intervention and a control where I didn't get that effect. And so the point wasn't, we figured it out. The point was, shouldn't we be doing things like this? Shouldn't we be thinking about how to leverage the spatial geography of these health risks and turn them into new kinds of questions, new kinds of interventions? Now, as we get to the home stretch here, a number of concerns, especially on the infectious disease side, are much bigger than humans. And I think this is probably true in toxicology too. But you know, we have a meeting with the state's veterinarian at the end of the month, because if you look at avian flu or you look at a couple dozen other things, where there are animals in the mix, right? These sort of zoonotic risk areas for us. We have the ability to understand animal health too. And if you have a concern where there's something that's not human, but is human relevant, we can start working on those kinds of things. Obviously, you know, we have a slaughterhouse down the road here that contributes directly to our sewer system. So we have to keep track of things that are in pigs as part of our work. And when the livestock show is here, that'll all be going into the Derek Guthrie treatment plant. You know, so we have to sort of look at what is that opportunity. It's either a terrible thing because I got all these other animals to keep track of, or it's a great thing because I should be looking at some of these other animals and maybe their health status or their health risk. And so, you know, I think we have this bigger opportunity, and, you know, it's going to take people with other kinds of training to help us really tease it apart. Then there was my favorite feedback from the EAC on Monday. And there is a YouTube video, that link, but people drinking sewage. So, you know, I've got a little attitude about this. Like, we have to, it's a great point that, especially as we move away from infectious diseases that disproportionately affect human beings and we move into these other environmental exposures, I think we need to be very clear about what we're looking at. And so the feedback I think we got was, you're not looking at an exposure. You're looking at something that is later, potentially, than an exposure. and may or may not be associated with an exposure. And it makes me think that part of the clarity that we need to achieve here is this platform has utility as a measurement of human health exposure, and it has utility as a measurement of environmental contaminants. They're two different things, right? If I wave an air monitor around in front of you, you'll say, you're doing environmental monitoring, and you are, right? If I get biomarkers out of your blood and urine, you'll say, you're doing human health monitoring, and you are, right? So we have a kind of a confusion here, and I think we got to clear it up, is the truth, because I actually think it has utility in both cases, but it's not good to be sloppy about how we're talking about it, right? So we're going to, with a renewal application, we're going to be crystal clear about anything we're doing in this realm. And on that happy note, I want to thank everybody who did all the work. And really, this is you heard whatever it is, it's 30 publications in the last 3 years. It's 2 government reports. It has been a lot of people at the University of Louisville. Really proud to call them my colleagues. We've worked with just about every core that's relevant. And then most recently with the CIEHS cores, it's just been a fantastic group of people. And I'm really looking forward to what we can do next. So thank you. Questions? Financial questions?
Is there any wastewater treatment system to remove the contaminants?
So we have 5 wastewater treatment facilities in Jefferson County, and their job under the EPA Clean Water Act, not the Clean Drinking Water Act, the Clean Water Act is to remove a list of criterion contaminants from the sewage, and then it's discharged into open waterways in our community. Some goes into the Ohio River directly, some goes into Lloyd's Fork, some goes into Cedar Creek. So yes, it is all treated. The thing to watch out for is whatever is in the Clean Water Act is what is treated. And so these emerging contaminants like PFAS, they're not required to remove PFAS, right? And one of the problems with forever chemicals is they bioaccumulate. And so, whatever Pittsburgh started doing to the Ohio River, we're a couple of hops down of wastewater treatment plants that have been discharging sewage that contain pharmaceuticals, that contain PFAS and PFOA, right? So it's important to not get too comfortable that we say, well, we have wastewater treatment, because we have limited wastewater treatment by definition. And it may take a long time to figure out what are the other things that really should be removed from the water before it's pushed back out into the cycle. There are, if you go to a wastewater treatment plant, there are sort of layers of digestion, to break things down. There are layers of filtration, and often a UV light is used to sort of finally sterilize, nasties that don't otherwise die easily. That's it. It's a big sort of digestion scheme. And in the case of Louisville, and our largest wastewater treatment plant, and this isn't unusual, we'll take those solids, whatever's dried out, which is sort of nitrogen carbon rich, turn it and call it fertilizer, Louisville green, which you all made. And then we give it to farmers and put it on the fields. So the number one concern, by the way, on that is to the extent that these PFAS compounds are in those biosolids, we're putting them on the fields, they're getting into the food, and they're returning to the water system, and then they're coming back out again, right? So we are just sort of, it's a closed loop system, we're just intensifying every cycle. So the state's highest priority with PFAS is to break the chain of sending this stuff out to the fields. Now, then what do you do with it? And that's a whole other problem.
This is so great.
I'm going to make a convert out of you yet.
It was awesome to see all this work, and like to go to your point about kind of what you were saying, exposure estimates and individuals may not be like reflecting what we're exposure estimating in population sources. And I just want to like think about that all together. Like in some ways, I think that's like probably one of the most detrimental public health messages we can do, saying that like population level exposure estimating isn't reflective of individuals, which is, it goes against public health, I think. And so like I think we should put in like an R01 or something so that people don't lose sight that like population level exposure estimates can reflect the individuals. Like I think at some point between Jon Snow and us, we lost that understanding, and like what do we need to do as scientists to like reconvince people that of that, and like so I don't know if you all have thought about that.
I mean, I've thought about it a lot, because we're in the school of medicine, and we think about clinical interventions, one patient at a time. Our clinician friends are happy to say, you can show me all this data you want, but I got to deal with this person, and that doesn't speak to this person, right? And so we all know the nature of the problem, but I think that's why having these complicated schemes where we have to acknowledge there's things that we don't know in the individual biomarker data. Like, for example, I don't know how long you've been exposed to something, right? And so a lot of this, I've oversimplified it. Like, oh, you got colorectal cancer, here's these metals, that's why you're like, maybe you were exposed 20 years ago, right? So how do we have a conversation about what we don't know that makes it hard to look at any given patient and apply that population data? So the population data includes time, right? That often clinical data doesn't include. And how do we talk about time? Because we don't talk about it. And then if we had, I look at the all of us cohort and say, there's just a lot of work to be done there, right? I mean, if you want to tell me about environmental exposure, we're really going to have to look at not just the stuff the satellite took a picture of last month, but how long did you live there, right? Where'd you work 10 years ago? I mean, like, it's not as simple as I got your geo coordinates and we're going to answer this question. So I know I'm not answering your question directly, but we do have a date with bringing these two things together. And I think this is a good way to do it, because we can highly describe small groups of people, individual patients, and we can attempt to put them in a bigger picture and then try to articulate all the other factors that we still don't have a good handle on.
Thanks.
Does it work? We got to continue R1, whatever you want to do.
Data, like, people don't have this problem with like monitoring air pollution, that reflecting, population exposure to air pollution. So like, there's like a disconnect for me with the wastewater. Or why is it so hard for people to realize population exposures are indicative of what individuals are being exposed to? So but
It's hard, bro.
It's hard to.
Oh, there he is.
Problem, Natalie, that you're probably not aware of. Just assuming that Emory, they do LC and GCMS both.
I think he's making these from these samples.
Okay, so particularly
Two different platforms.
with the LC, I think we'll see what they get back, but a lot of have some human samples on their platforms. And you get back on LC with 2 columns, you get back approximately 25,000 peaks in blood. 2 or maybe more or less, not all of them are named. So some of them are biomarkers of exposure. PFAS to get an LC. But others of them are metabolites. And some of these metabolites, I think they're going to be in the sewer because they come out in urine and food. And some of these metabolites are metabolites of disease or biomarkers of disease. So you can actually, it's going to be require a whole new kind of epidemiology, but you can actually do association studies between exposure and disease theory using this approach. That's going to require development of techniques. So like, I guess you got one one site at one time point, and you count that as a person, and then do the other sites as like a person, or you do multiple time points, how are you gonna do it? It's gonna require methods development. Other question.
This is from Dr. John Wyatt, Jr. How does this method factor in large influxes of people here for only a few days, such as under removal, derby, warning, and fest?
That's a great question. So among the many problems that we have are the fact that people are not simply glued to their toilets and that we know who they all are. So yeah, I mean, we have early in the pandemic, I don't know, San Diego called or whatever, and like they're in a situation where their entire population quadruples on the weekend. And I mean, it's just impossible to do COVID work, you know, when you have those kinds of crazy distortions. You couldn't make any conclusion about what the situation was because of the influx and outflow, right? So we have those events, they're rare, but we do have 1 million tourists who show up and they get their bourbon and they do that. I mean, they're there and they're coming in and they're going. You know, so this is just one of these background challenges that we have. As we are in residential neighborhoods, working in catchment areas, I start to breathe a little better. Lots of Airbnbs, so maybe I shouldn't. But, you know, having a sensitivity, it used to be, do you know where the hospitals are? And that was a good question. Like, do we know where the hospitals are? Because they have people with COVID in it, right? Like, so we would, we want to use the map data to our advantage, and we want to be thinking about things like that. So it's important. And I'll tell you from a national perspective, it's a great new, different opportunity, right? Like, how are things moving across the country? We can look at cities that have a lot of movement of people. There's airport surveillance in the United States. So the US Department of Defense pays for civilian aircraft surveillance right now, because we certainly want to understand what's coming into O'Hare and Newark and whatever. And that is one way to do it. Only works on long haul flights, really. But, you know, so it really matters that we think about what are the questions we think we can answer and what are the things we're trying to protect ourselves from, you know, to adjust for or otherwise treat. So it's a great question.
I had a nice presentation, thank you. So with respect to the environmental contributing factors for pollution, my kind of concern of the observation, like the kind of pesticides, herbicides, or the fertilizers applied to the lawns, everybody's yards, right? That can be a big, will that contribute to the contribution to contaminating the wastewater? It is like the health impact, right? It's definitely less addressed, right, by the researchers and the public policies. No, I talked to my neighbor actually. After every time after we spread the pesticide to the grass, and even dogs don't want to run into the grass. So let's think about that. Also impact to the resident, right? Every household, right? And then to the groundwater, right? Also probably run into the wastewater and the sewage system. I'm not sure. So can you?
Well, this is where Chris's good suggestion that we start looking differently at combined sewer systems from dedicated sanitary sewer systems. It can help us. I mean, it's not going to be perfect, but it can help us detangle these two things, right? Because in the case of combined sewers, the stuff running off the lawn is running into the sewer system and will be seen in the sewer catchment. The stuff that's outside the watershed is not running off the lawn into the sanitary sewer system. And so we have an opportunity to see perhaps what just people were exposed to in that for those pesticides or whatever it might be. But it is a mess. I mean, there's a lot going on here. And I think the most important thing we can do is work on what is the whole list of things so that we're at least being honest about the limitations. I happen to think it's additive to other interrogations. I don't think this is a thing to do. Like, I don't think we should build a center of excellence on wastewater and say, we figured it all out. I think we should be strategically using data like this to help clarify things. I mean, the point on if it can help us, you know, bridge population versus individual case data, that's great, but it's never gonna, in my opinion, answer something entirely definitively at the community level. And But thanks.
Great job. So a couple of comments. One is back to your V diagram.
Yeah.
So this sort of thing is useful at the top, right? So community exposure and public health, general public health warnings and such. Very fits right in. And then the other thing is with the events, right, where you have an influx of people. If you're monitoring for disease through that and you see a spike, well, then you know that it's been introduced into you. Once again, we have a public health advisory. So just a couple of really useful things once we get some of this really.
That's great. Thank you. We did it. Everybody satisfied? Awesome. Thank you so much. So much for your help.